Mariaelena is currently a postdoctoral researcher at the CTOX lab at the School of Science and Technology at Nova University of Lisbon, Portugal. And when I looked at her career, she's an expert on zooplankton and marine invertebrates. And her most recent research focuses on the bioactive compounds you find in marine invertebrates, specifically polychaetes. And today she will show a little bit of the work she has been doing in this field. So Mariaelena, thank you for taking time out of what I know is your very busy schedule to give this talk today. The digital podium is yours.
Thank you. First of all, I want to thank Ronald for inviting me today. It's a great pleasure and honor to be here with you today. So as Ronald said, I'm a marine biologist specialized in ecotoxicology and ecology ecotoxicology. And since 2019, I'm a postdoc researcher at CTOX lab. So just share my presentation with you. Okay.
Yep, I can see it.
Okay, nice. Let me, sorry. Let me start again, please.
No problem. Take your time.
Oh, I think that, okay. Okay, so can you hear the presentation?
I cannot see the presentation. I think you have stopped sharing, so you should.
Sorry.
No problem. Just
because okay, I have some problem with the presentation. So okay.
Now we can see
now?
it.
Okay, sorry. Let me put. Okay.
Perfect.
Okay. Sorry about that.
No worries.
So just give me 1 second more. So okay. As Ronald said, I'm a postdoc researcher at CTOX lab. Our lab leader is Professor Pedro Costa, and we are located at the Faculty of Science and Technology of Nova University of Lisbon. So our lab is a multidisciplinary group aims at understanding how marine fauna responds to chemical aggressors. So this includes investigating how marine annelids acquire ability to produce toxins. So one of our goals is exploring the potential of marine invertebrates in terms of bioproducts. And we are working with some kind of lot of marine vertebrates, such as marine annelids, anemone, gastropods, cephalopods. So toxicology and biotechnology involve the integration of multiple domains of expertise and methodologies, such as microanatomical and histological characterizations, also bioproduct extraction from marine invertebrates, bioproducts such as pigment on toxins that we also test on aquatic models, and also multi-trait phylogenetics and omics. So today I want to share with you some of our studies, and my presentation is titled Uncovering Toxins from Marine Worms, a Revealing Tale. The first research that I want to share with you was developed by Iris Ramos. That she was a bachelor student, and her study was already published in the journal Toxicon X. So the objectives of this work were evaluate the toxicity of putative toxins from two distinct Phyllodochida annelids, Aedes diversicolor, and Glycera alba. So Phyllodoxida is considered as one of the largest order within the Polychaeta and represents a group of holopelagic organisms that is distributed through a wide range of environments, spanning from shallow marine substrates to the deep sea habitats. Also, many Phyllodoxida are active predators, and some are known to secrete toxins as part of their feeding strategy. So Aedes diversicolor belongs to the family of Nereididae. It's known as opportunistic predator, and it uses its proboscis, aversible pharynx, that has two jaws. So it preys with these jaws and on other invertebrate animals. On the other hand, Glycera alba, it's carnivore, belonging to the family of Glyceridae, also known as bloodworms. It's a burrower that also uses its proboscis as predation tool. And we can see to the video it's a very aggressive species that uses its proboscis to attack its prey, and then during the attack should be injected into the prey the venom or the toxins. So we went to the field. We chose two different bays, Seychelles and Alcochete bays, near of the Mondego, near to the Tagus estuary. So we start with the animal dissection, and the target organs were selected based on the predicted site of toxin secretion. So the glands, the proboscis, the glands, and also we dissect a part of body wall to compare the two organs. Also, these glands in Glycera alba were noted to be composed by four sacs, and each gland being individually associated to a chitinojole. Then we collected extracts from the proboscis and the body wall of the two Polychaeta, and tissue samples were homogenized in buffer saline, centrifugated, and we collected the extract, supernatant, very clear. So we exposed muscle gills to those extracts, and then we collect the gills and perform the COMET assay, the single cell gel electrophoresis assay, to determine the DNA damage caused by the extract to the gill cells. So as we result our results, we noted that the skin of the Aedes diversicolor and the proboscis of Glycera alba caused DNA strand breakage in muscle gill cells. And also we noted significant differences relative to the control. And also we noted significant differences between the two extracts of the proboscis of the skin of Glycera alba. So we can conclude that the extract of Aedes diversicolor do cause higher DNA damage in the gill cells was from the body wall. That means that probably the toxin that Aedes produced are used as protection against predators. On the other hand, Glycera alba proboscis exhibits higher toxicity. That means that the toxins that Glycera alba produce are used during the predation. So also these findings highlight the diverse ecological roles of venom and toxins in different species, serving both defensive and predatory functions. So we went just a little bit deeper in the study of toxins from Glycera alba with this master project with the of Sonia Campus that was published in the Toxins Journal. So we know that the venom of Glycera tridactyla contains glycerotoxin. Glycerotoxin is a high molecular mass neurotoxic glycoprotein and also contain homologs of proteins that are known from scorpioniform fishes. So we aimed to investigate the presence of glycerotoxin forms in the worm-secreted proteome, as well as other bioactive of interest. So starting with microscopy, that is not was included in the paper, but the histology allowed for the identification of specialized glandular tissue and to confirm that the venom was produced only at the proboscis, since this was the only place where venom glands were found. So we can find the venom gland, and also we observed the connection between the venom glands and the venom delivery apparatus. Then, through the SDS page, the proteome was fractioned to simplify its complexity into 4 fractions. And the fraction proteins were compared with the described molecular mass for toxin known for other Glycera species, suggesting 7 possible peptide matches in Glycera alba based on molecular mass. So protein separation of fractioned extracts revealed further evidence for venom secretion. And the higher molecular weight fraction discloses important differences between the extract from the proboscis and from the skin, and that can be associated to the presence of toxins in the proboscis extract. Furthermore, such proteins may contain cap domains and bear neurotoxic properties, such as cysteine-rich proteins or CRISPs. CRISPs are very common in animal venoms and poisons, from snakes to gastropods. Then the analysis of proteins and peptides by mass spectrometry provides a total of 165 putative proteins with a positive identification against the customized database that include toxins and venom-related proteins. Of these, 63 proteins were only identified in the proboscis, 45 were only identified in the skin, and 57 common to both organs. Then glycerotoxin, glycerotoxin was present among several toxin candidates. In the proboscis, two potential glycerotoxin homologs with a more robust coverage were found, together with a higher number of matching peptides. In skin extract, we only could be matching against one glycerotoxin. Then gene ontology analysis disclosed a pattern of proteins related to toxin and other venom components. So I show you the top 5 terms of proteins identified in the crude extract of Glycera alba, skin, and proboscis. So we identify differences between the two different extracts, and the GO-term calcium channel regulatory activity was highlighted in the proboscis extract and was related to glycerotoxin and probably other toxins such as crisps. So we can conclude that our study indicates that glycerotoxin produced by secreted by glycerotoxin proboscis, and also the combination of proteomics with toxicity testing validate the evidence of toxin and classification of Glycera alba as venomous animal, like its congenerics, and however, it must be noted that the current study it was a primary approach to a no proteome. So with my presentation, what just emphasize that the relevance of marine annelids in bioprospecting for novel proteinaceous bioactives for potential biomedical applications. And that's it. I just want to thank you. Thank you for your attention.
I'm clapping for you, Mariaelena. Thank you.
Thank you.
For your interesting talk. If you unshare your
okay.
screen, then we can see if some people have questions or comments.
Of course. Give me just.
There we go. So people, if you have a question or a comment, you can either just activate your camera and speak, or type it in the chat if you're shy. I can start off.
Please, easy ones.
I will do an easy one. When you looked at the first study you did, where you compared the two phyllodocytes, do you have any idea what kind of proteins could cause the DNA damage? Do you have a hypothesis for what causes the DNA damage that you observed?
What kind of proteins?
Yeah, I mean, I would assume you need an enzyme or something, a nuclease, or.
Yeah, with comet side, we can't see what kind of proteins or toxin can damage the cells. So we also just have a total results of the damage, and also just we can say that the extract of the proboscis and the extract from proboscis of Glycera and the extract from proboscis of the body wall of Edista caused the higher percentage of DNA strand breakage. But I can't say how toxins specifically.
Okay.
Yeah.
Let me see. There's a question from Matthew Beatty. He asks, is it known at what stage of their life cycle for Glycera alba that the venom cells occur? Are they present in larvae?
I can answer to the question. I don't know if the venom cells occur in the larvae. I really don't know.
Do they have trochophore larvae? Do you know? Because I imagine if they have typical polychaete trochophore larvae, then I would bet a salary on it that they don't have those venom cells, because the cells that produce the venom are only present in the adult morphology. You don't see that in trochophore. So but it's an interesting question.
Yes,
when in ontogeny these cells emerge. We don't know. I don't think there's anything known about that.
I don't know. I really don't know. Thank you. Thank you.
Another question about morphology. So when you did the histology, you showed, the jaw associated with the venom gland. Did you at all see these pharyngeal lobes that have been described for Glycera tridactyla? Because in the paper where they described glycerotoxin in, I have a vested interest because I'm a co-author of that study.
Yes,
we found that you have the big glands, but then you also have these flaps, these little lobes associated with the glands, they're separate from them and they express the glycerotoxin. So I was wondering if you in this species saw that as well.
No, we can notice that we observed the connection between the venom glands and the venom delivery apparatus. Yes.
Yeah. Yes.
But I can see what you are describing.
I think somebody needs to do a good description of this, because it is a complex system. What are the real glands? You know, they have the muscles around it. It looks like a gland. It is definitely the reservoir connected to the jaw.
Yeah.
But then there is this separate anatomical structure that expresses glycerotoxin. And we have data for the other species that you briefly mentioned, Glycera phalanx and dibranchiata, where they have these lobes as well. And not only do they produce, not all species have glycerotoxin, but all the other venom toxins are also expressed in that structure. So somebody would need to do a nice, detailed, three-dimensional reconstruction of this gland system, because the textbooks have it wrong so far. It's more complex than the textbooks say.
Yeah, it's very complex. Yes, it is. But probably, well, we can do this.
Yes, exactly. Well, that's for the future.
Yeah, in the future, you know. Yes.
Anybody else? Any other questions? Or else, talking about glands, it may be a very good bridge to the topic of the next speaker. Thank you very much, Mariaelena. It was a very